Hello. Welcome to today's review session, AP Bio review session on DNA and protein synthesis. So today we're going to be focusing on the structure and function of DNA as well as how the DNA is transcribed and translated into proteins. So first we need to discuss Hershey and Chase in 1952. So you need to remember that a bacteriophage is a virus that infects a bacteria, and it is composed of DNA and protein. And Hershey and Chase did some experimentation to try to determine which part of the virus was actually infecting the bacteria and was actually being passed as the genetic information. So they labeled the protein in the bacterium with a radioactive label, which we will call S in this particular diagram, and the DNA with a radioactive label P. So as you can see in the first experiment, they took their radioactive sulfur and they added it to the protein, and the bacteriophage infected a bacterium, and then they condensed all that down into basically mush, and then put it in a centrifuge to let the solids separate from the liquids to determine where that radioactive label was found. And they found that the pellet, which is the bottom part, which contains the bacterial cells and the contents, did not contain that radioactivity. It was actually in the liquid. So that would have still been left with the shell of the virus. So then they did another round where they labeled the DNA with the phosphorus, the radioactive phosphorus. Same idea, went through the experimentation, mushed it all up, condensed it down and then put it in the centrifuge, and here we see that the radioactive label followed the actual DNA and the cell contents of the bacteria. So therefore, the conclusion was that the DNA was what entered the infected bacteria, so DNA must be the genetic material that is passed. Then we have Edwin Chargaff in 1947, who basically did experimentations that gave us the basic complementary pairing rule, in which we now know that the percent of adenine in DNA is pretty equal to the percent of thymine, and the percent of guanine is going to be equal to cytosine. So that tells us that those two are the different pairings of the actual nitrogenous bases in DNA. So adenine will pair with thymine so that the amounts of each of those is pretty much equal, and then guanine matches with cytosine. So again, those percentages are equal. So let's look a little bit at the structure of DNA. Watson and Crick and Rosalind Franklin are the scientists that are credited for determining this overall structure of DNA. Watson and Crick utilized a lot of the information about the actual structure and the base pairing. Rosalind Franklin's work with her X-ray diffraction of the DNA helix is what gave us the information that it was a helical form. And without that, Watson and Crick would not have been able to come to the final conclusion of the DNA structure overall. But we do know, since they put all that work together, that DNA is a double helix. The backbone of DNA, which is basically along the sides, is going to be composed of a sugar and a phosphate. So right here we can see the sugar is in blue, and with DNA it's the deoxyribose sugar, and then there's a phosphate group that comes off of each sugar. And they are connected to each other with covalent bonds. And then the rungs, or the interior part of the DNA molecule, is made up of the adenine bases, which we see here. And those are either going to be thymine, adenine, cytosine, or guanine. So again, the four main bases are adenine, guanine, thymine, and cytosine. We can separate them into two main groups. The purines, which are going to be your double ring structures, are the adenine and guanine. So a purine has actually two ring structures. And then the pyrimidines, the thymine and the cytosine, are going to have a single ring structure. So a pyrimidine is single ringed and a purine is double ringed. Okay? So the pairing is one purine with one pyrimidine. So adenine will match with thymine and vice versa. And cytosine matches with guanine and vice versa. All right, on to this, continuing with the structure. Again, we know that it's covalent bonds that hold the backbone together, the phosphate with the sugar. But in the middle, where the nitrogenous bases are going to connect, it's going to be connected with hydrogen bonds. So these bonds are very weak, if you remember from the biochemistry review, and so that allows them to be able to be broken very easily, which is very important when we need to replicate the DNA or transcribe and translate the DNA for protein synthesis. So the hydrogen bonds are going to hold the molecule together much like a zipper, allowing for it to be broken apart if needed. We also need to remember that the structure of DNA is anti-parallel. So one strand goes from the 5' end to the 3' end. So that's going to be on this picture here in the middle. We can see that the 5' to the 3' is on the left-hand side, and then the right-hand side over here is 3' to 5'. So they are anti-parallel. So the 5' end has the phosphate group extending off of it. The 3' end does not have a phosphate group. Okay, so 5' has that extra phosphate group kind of hanging off of it. That's what makes it 5'. All right, and DNA is, the amount of DNA, especially in animals like ourselves, humans, is so extensive that there's no way it would fit in each of the cells, and it does contain all the genetic information, so it needs to, but so we need to have some help to get it to fit inside the cells, and so that is done with the help of histones. Histones are little proteins that the DNA can wrap around, and so the DNA will actually wrap around a group of histones and will form these little nodules called nucleosomes, and then the nodules are able to kind of bunch together to form our chromosomes. So that is how we are able to squeeze all that DNA into a chromosome, which will then fit inside the nucleus of a cell. So you can see here we have the chromosome being formed by this looping and scaffolding of these nucleosomes being wrapped because of the wrapping around those histones. So if we compare prokaryotic DNA with eukaryotic DNA, they are similar, but there are some differences. They both are double-stranded DNA, but in the prokaryotic cells, it's just a circular one chromosome. It's just one chromosome that's circular in the middle of the cell, and it's not in a nucleus, it's just in the cytoplasm, whereas eukaryotic DNA is going to be linear, and it's going to usually result in more than one chromosome. Typically it's an even number because we have a diploid number, and they are found in the nucleus. Okay, and because there's only one circular chromosome in prokaryotic DNA, there's no need for histones. Okay, whereas in the eukaryotic DNA, we do need those histones, those proteins for the DNA to wrap around it to form that chromatin and chromosomes to be able to fit. So let's talk a little bit about how DNA is going to replicate and make copies of itself. So remember that prior to any cell division, we have to make copies of the DNA so that the genetic information can be passed to the daughter cells. So in order to do that, it is a semi-conservative process, which basically means that the new molecule is going to be part of the old and part new. So it's going to actually retain some of the parent DNA in the new DNA. So we can see here that and we have a separation of the strands. If this is our parent DNA in the darker blue, the hydrogen bonds are going to break apart between the nitrogenous bases, separating the two strands apart from one another so that they can each become a template for the new strands that will be formed. So one side will be part of a new DNA molecule, and the other side will be a part of the other DNA molecule. So they will form the template so that the daughter DNA can actually complement next to it. So here are the major steps of replication. First step is we're going to have an enzyme called helicase, which will help unwind the DNA at the origins of replication, the points of the DNA that are going to be replicated. So the helicase unwinds it. Then we are going to have the initiation proteins. They're going to separate the two strands, so separate those hydrogen bonds, and that will form a replication bubble, so an area that's going to be replicated. Then the primase, another enzyme, is going to put down an RNA primer to start the replication. So we can see that in this picture here. We've got primase coming in over here. Here's where the strands have been split like a zipper. Okay, the helicase has come in to unwind it, and we've got a bubble, the replication bubble starting, and the primase has come in and put down an RNA primer to prepare for that replication. And then DNA polymerase 3 is going to add the complementary bases to the leading strand. So that's going to be the leading strand is going to be made from the 5' end to the 3'. So let's take a look at that in this next slide here. Okay, so we can see here that we're going to be building, we have the polymerase right here, and this is the nucleoside coming in. So the nucleoside is coming in so that this is the 5' end of the new complementary strand that's being built, and then the 3' is down here. So it's being added to the strand. This would be the 3' end of the template strand, and this would be the 5'. Okay, so because they're going to be anti-parallel. So the 5' is going to be built on the leading strand, so the leading strand is actually going to be 3' to 5', and it's going to be built in anti-parallel fashion. Okay, so that would be considered the leading strand. And that is done with the help of the DNA polymerase, which again is another enzyme. The lagging strand, which grows in the opposite direction, the 3' to the 5' end, is done by the addition of Okazaki fragments. So it's actually done in a fragmented method. So let's go to that. Oh, this is just showing you again replication of the leading strand and how that works. The RNA primer, and then we've got the DNA polymerase that's bringing in and building the complementary until we get all the way down to where we need to stop. Okay. Okazaki fragments are going to be used to build the lagging strand. So you can see we have one fragment here, fragment 1, that's going to be put in, and then we're going to get another primer, RNA primer, and then we're going to get second fragment put in, and then we're going to get another primer, and then another fragment would be put in. And so there's primers that are put in between the Okazaki fragments until finally the DNA ligase, which is shown here, is going to come in and actually seal remove the primers and seal the actual fragments together. Okay, so but you can see that it's going from the 3' end to the 5' ends. It's going in that direction. So opposite of the leading. All right. So the DNA ligase actually seals all the fragments together there. All right. So here we can see this is the 2 ends. We've got this, the leading strand on this side, and it's going from the 5' end. This is the origin of a replication on this side. So you've got a continuous building of the replication happening on the leading strand going that way, from the 5' to the 3'. And then on the lagging strand, we're going from the 3' to the 5', so we are going in the same direction, but we are being built in fragments. Okay? The little fat fragments that are being built. And it's going 3' to 5'. All right. So leading goes 5' to 3', lagging goes 3' to 5'. All right, now once the DNA is being built, we have to have some proofreading and repair. So the DNA polymerases proofread as the bases are added. So as the DNA polymerase is adding the bases, they are proofreading those. If there is a mismatch repair, special enzymes will fix the incorrect pairings if there's a mismatch repair. If there is a nucleotide that needs to be repaired and an actual nucleotide needs to be excised out, then there are enzymes called nucleases which will cut out the damaged DNA, and then the polymerase and ligase will fill in the gap. So we can see that in this next slide. And this is an example of a nucleotide excision pair. So we've got a problem here. So the DNA polymerase is going to, that part's going to get cut out, and the DNA polymerase is going to redo it, and the ligase, you can see right here, is coming in to seal it back up. So pairing errors happen 1 in 100,000 nucleotides. Complete DNA error would happen 1 in 10 billion. So pretty rare, but they do happen. Now, there is a problem at the 5' end. So when we're making that lagging end, those fragments from the lagging strand, at the 5' end, we have, of course, that phosphate that sticks out. And so what happens is as we are replicating, it ends up becoming shorter and shorter because there's no way that polymerase, the DNA polymerase, can add, can finish off that end of the 5' because of the phosphate being there. So it can only add nucleotides to the 3' end. Okay, so we can see that at this point here, because this is the DNA primer, and so that's going to be left blank. So now when this goes to replicate again, so that has nothing to it, so it'll start, the replication will now only go to this point, and you can see that we now have this part ending. So as you continue on, the daughter molecules become shorter and shorter. So over many replications, DNA strands actually grow shorter and shorter. So to help with that, so that we don't start having problems with actual important information being left out of the replication, there are these things called telomeres. So telomeres are repeated units that are short nucleotide sequences. This tends to be the sequence TTA, GGG, and they're at the ends of DNA, and they're repeated numerous times, and they're kind of like a cap at the end of the DNA. And so it helps to keep the DNA from eroding away genes that are necessary for the cells to do work. Because once we get to that point, those cells will no longer be able to function properly, possibly, so they will end up dying off. So it is a process that shortening of the DNA is an aging process, so that is what allows and leads to aging. But the telomeres are there to keep it from happening rapidly. So telomerase is an enzyme that adds telomeres, and this happens in germ cells, which are the cells that become our reproductive cells. And also cancer cells will have excess telomeres, allowing them to continue to grow and invade. So you can see in this little picture here, these are mouse chromosomes, and the telomeres have been stained orange at the ends of these chromosomes. So you can see the little orange pieces. All right, so the flow of our genetic information goes from DNA to RNA to protein, and this is known as the central dogma. We know that DNA is the genetic, holds the genetic information, but to get it into action, it has to be transferred to RNA so that proteins can be made. So the actual transferring of the transcription, or of the DNA to the RNA, is called transcription. Okay, so that is the initial part of the protein synthesis, in which we have to get the code from the DNA onto the RNA. And then translation is when the RNA takes that code and actually puts together the protein. And the site of translation is going to be at the ribosome. Transcription, on the other hand, takes place in the actual nucleus. And so gene expression is the process by which the DNA directs the synthesis of the proteins. Okay, sorry about that little pause. So we were talking about, again, the central dogma is the DNA to RNA to protein. Okay, so in prokaryotes versus eukaryotes, a very similar process. We have to go from the DNA to the RNA to the protein. So if the only major difference is that there's no nucleus, so with the in the prokaryote, so there is no need to come in and out of a nucleus to get to the protein, whereas in our eukaryotic we do have the nucleus, so we actually will have that transcription process take place in the nucleus. Also, there tends to be extra information in a lot of the eukaryotic chromosomes and DNA, so there will be a pre-messenger RNA, which is prior to the removal of that excess information. One gene equals one polypeptide. Remember, polypeptide is multiple amino acids together, so one gene is one polypeptide. Remember that DNA is going to be composed of nucleotides as their building block. The same is true for the RNA. Differences between the two is that DNA is a double strand, RNA is single strand. RNA has ribose as the sugar, DNA has deoxyribose. As far as the bases are concerned, they're all the same except that RNA does not have thymine. Instead, RNA replaces thymine with uracil. Okay, and then DNA is the template for the individual, whereas the RNA is the helper that moves the DNA information to the protein being made. So the RNA plays many roles in the cell, and these are some of them. The main ones that we like to talk about are the pre-mRNA, which is the newly transcribed and non-edited messenger RNA once that happens in the first part of transcription. Then we have the messenger RNA, which is the edited version that carries the code from the DNA to the amino acids. We have the tRNA, which is going to carry a specific amino acid to the ribosome based on its anticodon to the messenger RNA codon. They have to be complementary so that the amino acid is the correct one. The rRNA is primarily what makes up the ribosome, where we have the protein synthesis of translation take place. Then we have snRNA, which is small nuclear RNA, and it's part of a spliceosome. It has some catalytic roles and structural roles. We have the spIRT RNA, so the SRP RNA, which is a signal recognition particle that binds to signal peptides, and then RNAi, which is interference RNA. It helps regulate the molecule. So here's an overview. We have up here the genetic code, the DNA molecule in the box here, and you can see that certain parts of the DNA are going to code for different genes. The DNA template is, if we were to look at it closer, is down here, and it looks like a ladder, and then that's going to split apart, much like it did for the replication, so that transcription can happen in which the messenger RNA can be built as a complementary code to one side of the RNA of the DNA, and then that messenger RNA, once edited, will then move to the protein where the amino acids are actually going to be built based on the codons. The codons are every 3 bases, so a triplet base is what represents one codon, which represents an amino acid. And this is an example of a codon chart. There are 20 different amino acids in nature, and there are 64 different codon combinations. There are usually more than one codon that will code for each of the amino acids. That's what the redundancy means. So there's usually greater than one that will code for the same amino acid, which helps with some mutations or that might occur from the major. A reading frame is groups of 3, so it has to be 3 bases to be able to be the correct amino acid. And this code is universal, so all forms of life use the same code. It's just a matter of the putting together of the different polypeptides as to what different proteins are made and what organism we get. So this is one of the universal parts of all life. So transcription is that first process in which we are going to get the code from the DNA and start taking that message to the ribosome. This happens in the nucleus. So the stretch of DNA that codes for a single polypeptide is going to be where the transcription will take place. We have RNA polymerase, which will separate that DNA strand so that the hydrogen bonds break apart, and will begin the transcription of the mRNA. And it is going to build itself from the 5' to the 3' end, so in that direction. And the key thing to remember is that when it is building its complementary, the only difference is that wherever there is an adenine on the DNA, it will match with a uracil in the messenger RNA instead of thymine. There will be a promoter at the start of the gene, so that's the start of the actual polypeptide, and then there's a terminator at the end, so it knows where to stop. So once we have that messenger RNA built, we then have to edit it because there are introns and exons. Introns are the non-coding pieces. These are the pieces that don't code for any part of the polypeptide that we're looking at. And the exons do. So exons exit. So I like to remember that by the fact that exon has the EX for exit, and so that's the parts that will exit the nucleus to go to the ribosome. The introns will stay in the nucleus, so they're not going to come with us. So the introns get cut out and the exons are joined together. This then moves to the ribosome, in which the messenger RNA, of course, is the message. So here's our messenger RNA down here in red. The transfer RNA is the interpreter, and it's the blue piece here. And then the ribosome, which is this big blob surrounding, is going to be our site of translation. Transfer RNA, this is what it kind of looks like all blown up. Okay. It's transcribed in the nucleus. It's going to be specific to an amino acid. It has an anticodon that is going to be specific to an actual amino acid. And so it's going to match or complement the messenger RNA codon. And if it complements correctly, then that amino acid that is hooked to that particular transfer RNA will be added to the polypeptide. So the anticodon has to complement the codon for the amino acid to be left behind for the polypeptide. On the ribosome, there are 3 spots that kind of match up with that anticodon. There's the A, which is the active site that holds the amino acid to be added. P is the site where the polypeptide kind of grows from, and E is the exit site. So the transfer RNA will kind of move down those sites. So it brings in, you can see down here, it's bringing in the amino acid to be added based on the codon, and then moves over so that it can be added and held to the other amino acids, and then once a new one comes in, it can then exit and leave. So again, just overviewing the central dogma: DNA, RNA, translation protein. So DNA, RNA, protein. DNA polymerase is involved in the replication as well as the, is involved in the replication of DNA. RNA polymerase is going to be involved in the actual transcription, and then the translation involves the ribosome there. Okay. Mutations, if mutations happen, they are going to happen at the DNA and the replication process, but the effects are going to be seen in the translation, in the making of the protein. So let's talk a little bit about some of the mutations. Mutations are any change in the genetic material of the cell. A large-scale mutation is considered a chromosomal mutation, and these usually always cause a major disorder or death. So this would be things like that occur because of non-disjunction. So during the mitosis or meiosis, the actual chromosomes not disjoining like they're supposed to, so you end up with an entirely extra chromosome, or you end up with a chromosome that is missing. So that would be an entire chromosome. That's a large change that's going to result in either death or a major disorder. Translocation, in which a whole chunk of the chromosome is moved to another chromosome and they are switched. Inversions is where a whole chunk of the chromosome is actually inverted and reversed. A duplication would be where a chunk is actually duplicated or repeated in the chromosome, or a deletion would be where a whole chunk is actually not copied and not made. So those are going to be involving multiple polypeptides, multiple proteins, and are therefore going to cause either a death to the organism or a major disorder such as Down syndrome, Turner syndrome, things like that. Point mutations, on the other hand, are going to be a smaller scale. They can have major effects, but they're also more likely to not have as significant effects. So this is where one base pair of the gene is changed. So a base pair substitution is where one of the base pairs is replaced with another. So a missense base pair substitution is going to be where that replaced base pair gives us a different amino acid. So instead of, say, valine, it codes for leucine, which may affect the overall chromosome, or I'm sorry, not chromosome, may affect the overall protein that is formed in the functioning of that protein, but it's not necessarily going to cause death. A nonsense base pair substitution is where that replacing of the nucleotide is going to actually cause a stop codon where it shouldn't be, or does not code for an amino acid. A frame shift point mutation is where the messenger RNA gets read incorrectly, so it forms non-functional proteins. So this is either caused by an insertion or a deletion, so where a base is actually accidentally repeated, which would mean it will be inserted, or an extra one is read or added in for some reason accidentally, or one is skipped and missed. So it shifts the entire codon so that from that point of the mistake, all of the amino acids from that point forward are going to be ultimately wrong and not correct. So here's some examples of those. So starting up here at the top, we've got the original DNA and RNA. So you can see the DNA template, the messenger RNA, that's coding, and it's matching up to the top DNA strand there. And then the proteins that would be the amino acids that would be coded. So methionine, lysine, phenidine, and glycine. In this A, we have a nucleotide pair substitution. So this is a silent one here because the substitution right here, where instead of an A, it says instead of the U, we get a instead of the C. So it should have normally been, where am I at here? So A-T-T-G. So it should have been a G. Okay, so but instead it was an A that was replicated for some reason and put in there. So now that A matches with a U. So it should have been a G matching with a C, but when we look at the codon chart, GGU is going to code for the same amino acid as GGC, and that's glycine. So in this particular case, there's no effect because it's still the same amino acid as it would have normally been. So that does happen, okay? However, in here, we have a missense in the second one right here. This is a missense because instead of the T, instead of the C that should have been here, we get a T. So now our messenger RNA is going to have an A instead of the G, and now we get a whole new amino acid. So it should have been glycine, and now we're getting serine. So that's going to give us a missense. It's going to give us a different polypeptide. The nonsense is where we get a stop codon or not a particular amino acid at all. And so that's what's happening in this particular one, where instead of the T here, we get an A. So now the A is going to match with the uracil instead of the A it would normally have matched with. So we get a stop codon in the middle of this polypeptide. So that's really there's no polypeptide that's going to form. So it's called a nonsense. On this side over here, on the B side, on this side, we've got frame shift happening. So in this first one, we get an extra A. All right, so an extra A is put in, which means we now have a U that's been put in here, and that's shifting all of these over. So now it's UAA instead of an AAG, because that U is added in. And so we get a stop codon. So here we've got, again, where we messed up the polypeptide that's not even starting. Whereas over here, in the second one, we get where the A is missing. So the A that was supposed to be there has been taken out. So now the U has been taken out. So it's changing not only this amino acid, which should have been phenylene, it's now leucine, and now the next one, instead of being glycine, is now alanine, and so on and so forth. So it's going to mess up all of those amino acids from this point forward. We can have a whole amino acid missing. So this has a whole, this last one down here causes an extensive missense because a whole codon was missing, a whole triplet. So now the whole thing, instead of having our lysine, we go from methionine to phenylene. So it changes the entire polypeptide. So those are just some examples of the mutation. An example of a disease caused by a mutation, a point mutation, is sickle cell disease. Sickle cell disease are where the red blood cells, instead of being the normal donut shape, are going to have this crescent moon shape, which is called sickle. And it's caused by the genetic defect, and it's carried by up to 25% in some regions of Africa. And so what happens in this is the normal hemoglobin codon. It should be from the DNA CTT, which then matches or complements with a GAA, which codes for GLU amino acid. In the sickle cell disease, the T is switched to the A, which now gives us a U here, and now it codes for valine, and that is the sickle cell hemoglobin. So the hemoglobin, which is the protein that holds the oxygen inside the red blood cell, is misformed. You can see the normal. Normal is over here. Sickle cell is right here. So they have different shapes, and therefore their functions are different. So this is where we talk about how shape can make a big impact on the function of the different molecules in our body. So normally our red blood cells look like these little donuts. They're easily able to move through these small vessels because they can squeeze and change shape. The sickle cell have these little crescent shapes, so they tend to get stuck and clot, and so it can cause major problems with that. All right, so let's do a few practice problems. Our first one is talking about DNA replication. So when DNA replicates, each strand of the original DNA molecule is used as a template for the synthesis of a second complementary strand. Which of the following figures most accurately represents the illustration, which of the following figures most accurately illustrates enzyme-mediated synthesis of the new DNA at a replication fork? So we know that it can't be A and it can't be C because neither one of these are showing the Okazaki fragments with the short little arrows. Okay. And then we've got to remember that the leading strand is going to be the continuous, and so that's going to go from 5' to 3', so that would be down here, and that's shown here. And then the lagging strand is going to go from 3' to 5'. So it has to go from 3' to 5' and 5' to 3', so it has to be, the answer is, this is actually not correct. It has to be D. Okay, so the leading strand is going from 5' to 3'. The lagging strand is going can only be added to the 3' side. So it's still going 5' to 3', but it's being added. Those segments are being added from the 3' onto the 3' side. So D is the correct answer. All right, sickle cell anemia results from a point mutation in the HBB gene, the hemoglobin gene. The mutation results in the replacement of an amino acid that has a hydrophilic R group with an amino acid that has a hydrophobic R group on the exterior of the hemoglobin protein. Such a mutation would most likely result in an altered what? A, properties of the molecule as a result of abnormal interactions between adjacent hemoglobin molecules. B, DNA structure as a result of abnormal hydrogen bonding between nitrogen bases. Okay, it's not going to have anything to do with the DNA at this point. That was already messed up. Fatty acid structure as a result of changes in ionic interactions between adjacent fatty acid chains. Nope, not going to have anything to do with fatty acids. Hemoglobin is a protein. D, protein secondary structure as a result of abnormal hydrophobic interactions between R groups in the backbone of the protein. This sounds like it might be right. The only problem here is it says secondary structure, and the R groups, there's R groups and secondary structure, and so that does not go together. Tertiary is where the R groups start to connect together. Secondary is where the hydrogen bonds form, so it cannot be D either. So it is going to be A. So the mutation is going to result in altered properties of the molecule as a result of abnormal interactions between the adjacent hemoglobin molecules, which makes sense because they do form these thick lines and stretches of molecule that are real thin strands in between the hemoglobin molecules in the cell, which is what causes that sickle form. All right, then we have a free response question here. Says the table below shows the amino acid sequence of the carboxyl terminal segment of a conserved polypeptide from 4 different but related species. Each amino acid is represented by the 3-letter abbreviation, and the amino acid residues in the polypeptide chains are numbered from amino acid end to the carboxyl end. Empty cells indicate no amino acid is present. So A, assuming that the species 1 is ancestral species of the group, explain the most likely genetic change that produced the polypeptide in species 2 and the most likely genetic change that produced the polypeptide in species 3. So if 1 is the ancestral, this would be what we would say would be considered normal. That's what they're basically kind of saying here. And we can see in 2, everything's the same except for number 4. We have lysine instead of valine. And then in number 3, it's actually all the same, but we're missing 2. So what might be the most likely cause? What's the most likely genetic change that produced the polypeptide in each of these? So best guess here would be that we have some kind of mutation taking place in both of these. Okay? So let's just take a look at our answer for A. So for species 2, a correct answer for the change would be mutation would be acceptable. You could have said a substitution, a point mutation, or missense mutation. Any of those would have been correct. You did not have to give a specific name of the mutation. And then the result of the change, you would need to explain what happens here. So you would need to say that that mutation then resulted in the amino acid change only at position 4. So you could see that that was the only change that happened. So we would say, you know, in species 2, amino acid number 4 was switched from valine to lysine, possibly due to a mutation. That would have been a perfectly fine answer. Species 3, you also could have said mutation. Again, you could have given more specific as far as point or frame shift, but that's not necessary. And we can see that there's no additional amino acids after valine, so probably ran into a stop codon. So the termination of the polypeptide after the valine at position 8 would be the explanation of what happened, and what caused it was either some sort of mutation or a stop codon. That was after the valine. Okay. The second part is predict the effects of the mutation on the structure and function of the resulting protein in species 4. Justify your prediction. So in 4, if we match it up to the beginning, it's got a change here, a change here, a change here. Like all of these basically are separate. So because all of these from one point on is a different amino acid than the original, the species 1, I would probably be inclined to say that this was from a frame shift mutation. So what's going to happen is it's going to obviously change the structure of the resulting protein, which will then cause it to not function correctly or as the original protein is supposed to. So let's just kind of see what what they want you to have here. So it says in B, predicted change would be, you could have put any of these. Protein may have a different structure and a change in function. Okay, that's exactly what I said. Protein may have a different structure and no change in function, or protein structure and function may not be affected. So you could have put any of those, you just had to back it up. So since I said protein may have a different structure and a change in function because of the different amino acids, justification of this would be because the change in the amino acid sequence of the protein starting at position 5 could alter the overall structure or local structure regions interfering with the function of the protein, which is ultimately what I would basically say. Anytime we change up the amino acids in the polypeptide, that's going to affect the overall structure of that polypeptide. Okay. If you chose to say that the protein may have a different structure but no change in function, some justification that would have fit that is you could have said a change in amino acid sequence alters the shape, conformation, folding, binding region, any of these things, but does not affect the critical function of the protein. If you said that the structure and function may not be affected, then your justification would have needed to be along the lines of change in amino acid sequence does not alter the protein shape and does not alter the function. All right, so in this case, they were, even though logic would say that when you have this many amino acids changing in a polypeptide, it's going to change the overall structure, which would then change the function. They actually allow you to say that that's not going to happen as long as you back up that you are saying that the change is not altering the shape and therefore would not alter the function. So it's all about justifying and answering all parts when you're doing the free response questions, making sure that you're answering all parts that is being asked, not only giving the first part to predict the effects, but also justify it. And as long as your justification backs up your prediction and it doesn't contradict the prediction, then in most cases you are going to gain points for that. Okay? All right, and that is it on the DNA and protein synthesis. Hopefully this was useful to you, and please thank you all for joining me. And if you have any questions or concerns as we continue prepping for the AP exam, please make sure you let me know. Have a good day.